Hi, everyone. This is Dr. Wiseman. Welcome to Chapter 26, Digestive System. The trick with a chapter like this is that we already have some fundamental familiarity. I mean, that may be true with all of the systems. The key is what else do we need to know, not just from a testing perspective, but also from a therapeutic perspective. It's not enough just to know what everybody else knows, or they wouldn't be paying you the big bucks to help them out. So we're going to work on not only some of the things you might already know, but we're going to try to take it to the next level, which is to understand some things that may not necessarily be known by the general public. So please take notes, just like this is a regular lecture, and let's get started. Two things that we are going to focus on are the processes that occur from the beginning to the end, shall we? In theory, following this lecture, you should be able to trace a piece of food, not only that food, but also the constituents of that food, the carbohydrates, the proteins, the fatty acids, lipids, etc. And you should be able to trace that process from start to finish, all right, from the mouth to the anus and what enzymes are going on. We're going to focus a lot on enzymes in this lecture. We're also going to focus a lot on the hormones. The thing about the digestive tract is that yes, we chew stuff here and we swallow it, and yes, we're going to digest it, quote unquote, and we're going to absorb it, quote unquote. But what's actually happening is a very, very coordinated system of hormone signals and functions of enzymes. So we're going to work on that. For instance, like we talk about amylase. We actually have two different kinds of amylase. Amylase will break down carbohydrates. This is one of the reasons why if you're really, really hungry and like you go to a Mexican restaurant or something, and you have that first nacho chip with salsa on it, it tastes so good. That's because your saliva is high in salivary amylase, and it will break down the starches, which normally don't taste very good. Um, starch kind of tastes like cardboard, okay? But when you break it down into the shorter sugars using salivary amylase, it will break it down, and that's what gives it its really, really awesome flavors when you first take that first few bites of chips while you're waiting for your entree. But after your entree, when you're just like done, you'll try tasting that nacho chip again. It won't taste as good because there isn't as much amylase. So we're going to be doing that sort of thing. Amylase is one of the ones we're going to talk about. We're certainly going to talk about some of the pepsin is one, and lipase, and some of these other things that are going to happen in these different areas. So this picture right here is actually really, really, really useful for you. Look to where the enzymes are located, like for instance, hydrochloric acid, pepsin in the stomach. In the duodenum, pancreatic amylase, which is different than the salivary one, lipase, trypsin, and these other small sugar, uh, uh, carbohydrate enzymes, as well as peptidase. So this is actually a really, really useful figure for you. So we have two fundamental processes. One we have is called motility of the digestive tract, and the other is chemical digestion and coordination of the digestive tract. So let's talk about motility. Most of us have some familiarity with this. It is the peristaltic motion. If you've ever swallowed a little bit too much and you can feel it going down to your stomach, you're feeling peristalsis there. If you hear your stomach grumbling, all right, or if you have somebody that you trust, you can lay their ear on their belly and you can listen to the kind of gurgling that's called borborygmi. It's one of my favorite words. That's peristalsis. That's the peristaltic motility. But we also have to break down material into smaller pieces to increase the surface area. That includes chewing and mixing with saliva, emulsification, and so forth. And that's all part of motility. On the second part, we have the chemical digestion. And one way we do that is through exocrine secretions. Now, what does exocrine mean? It means that there is some kind of secretion by a cell, but it goes into a biological duct. That is to say, a tube of some kind that then takes it to an exterior compartment. That's exocrine, as opposed to endocrine, where endocrine, the cell will release whatever it's releasing. Usually it's a hormone, and that hormone goes into the extracellular fluid. It doesn't go through a duct. It just diffuses out into the extracellular fluid and then to the bloodstream by implication. So things that we're going to secrete, certainly the enzymes that we were just talking about in the previous slide, all right? But we're also going to talk about hormones as well. It doesn't make any sense to secrete something down here when you don't have any material in it, or secrete something here when you don't have any material. In fact, it can even be damaging in some cases. So we only want to have secretions occur when there is a proper stimulus present, whether it's in the stomach or whether it's in the small intestine, large intestine, even all the way down to the very, very end. So it's a very, very closely regulated system, and we're going to talk about that in this lecture. Again, what are the things that we use to break down these things? Like, for instance, starches are polymers of carbohydrate, and we're going to break those down into smaller carbohydrates, maybe even as small as simple sugars. Proteins, we're going to break them down into their amino acids. Lipids and fats, we're going to break them down into the constituent components, fatty acids and glycerol. So we have physical digestion, that is mechanical digestion, chewing and motility. We have chemical digestion using enzymes and emulsifiers, etc. And then the third phase is absorption, getting that material from this external compartment into the bloodstream and or the cells. That's called absorption. As an example here, I have fats here, and fats are really, really interesting. They're a little more complicated than a lot of the other ones. What we do with fats is we actually have to make them create special structures that allow for absorption into cells and eventually into the bloodstream. Now, you aren't responsible for all of this, but I just wanted to show you this is an example of absorption where we're taking molecules, breaking them down, converting them into something that we can absorb, bringing them in and bringing them into an internal compartment, whether that's the bloodstream or the lymphatic system or in cells individually. Following absorption, we then have excretion. So material that either we have not been able to digest or do not want to absorb, or thirdly, this is a way of removing material from the body. We often think of urination as an example of excretion. We can also remove material from internal compartments and cells and so forth through defecation as well. It's a major excretory route. It's not just for undigested material. So we'll talk about that in a little bit. So we have mechanical digestion, chemical digestion, absorption, and finally excretion. And so we've talked about the sort of general functions, if you will, those first four major functions. For testing purposes and so forth, you're going to need to know digestion by each region. By each region, that is to say the oral cavity, the stomach, small intestines, and the accessory organs for the small intestine, liver and pancreas. Gallbladder is sort of in there too, but not a major role. In this figure, by the way, I want to point out something. It says the duodenum is separate from the small intestine, and some people will do that. I'm not too worried about whether it's in the duodenum or the overall small intestine, but make sure you take a note of that. Both of these things are the same thing, and what you don't see on here are the large intestines. We will have some material on that. Again, it doesn't get enough credit. All right, so we're going to work on things like what is primarily digested here. Like in the mouth, it's primarily carbohydrates. In the stomach, primarily proteins. All right. In the duodenum, certainly we have carbohydrates, we have fats, and we have proteins. So when you look at each of these enzymes, the thing to do is ask yourself, what's the basic function? What is it chopping up? Okay, sucrase, that's going to be a sucrose. All right, maltase, maltose, those are all disaccharides. Okay, peptidase, that's going to probably be a protease. All right, so what enzymes are excreted? What other processes that might be occurring in terms of absorption? All right. Certainly the mouth, what we're going to have is a lot of mechanical digestion. Same thing in the stomach. But once we get into the intestine, it's going to be more about absorption. Same thing's true in the large intestine as well. A lot of absorption occurs in the large intestine, but it's primarily water and ions and things along those lines. So again, break each region down and have a goal for knowing an answer to each of these three questions. Alongside the previous slide, that's kind of your study guide slide, if you will. This is not directly in the book. You could probably chase it down if you looked around a little bit, but this figure can help you a lot. So I would make sure that you take a little bit of time with this figure. We're also going to focus on the hormones. The hormones are hormones are hormones. So now in the last 5 years, there has been a revolution. I am not kidding you. A revolution in the understanding of digestive hormones, especially in the therapeutic realm of diabetes, obesity, and a term that you might not have heard before is called satiety, which is when do you feel full? Because if you feel full, you're not as inclined to eat. All right. It tapers off your desire to keep eating. And if you don't want to eat, then you don't bring in calories, you don't gain weight, and so forth. So I have 5 hormones here. All right. GIP, GLP-1, one called ghrelin, PYY, and CCK. We'll talk about CCK a little bit more down the line. But your goal is to use this figure and identify just a basic function. I'm not going to ask you to recite these from memory, but you should be able to say, look at a list and go, oh yeah, I know what GIP does. All right. It stimulates insulin secretion. It's also a satiety one. All right. There's that magic word. So those are kind of the things. Does it stimulate a secretion of some kind? If it's involved in satiety, I want you to pay special attention to this because that is the big thing right now, are these new drugs that are mimicking these drugs. All right. Inhibition of appetite and food intake. All right. Satiety. All right. That's just the same. That's just a definition of what satiety is. So these are super important. And so we're going to kind of step outside the book just a little bit with these. Again, I'm not asking for super thorough definitions, but you should be able to recognize a function from these. All right. And I have included an outside link for you. You can either copy and paste it from the PowerPoint, or you can use your phone or whatever, your camera, to go to this is the QR code for that URL. And this is basically how digestive hormones work. And by the way, this is a wonderful resource. You saw that figure in the previous slide. This actually kind of spells it out a little better for you if you want. I think it'd be worth your time. Basically, when we talk about hormones, you have a secreting cell, you have an endocrine cell. That's a cell that secretes a hormone. It releases it into the bloodstream or the extracellular space and then into the bloodstream, travels through, and then will bind a receptor on a target cell. That receptor can be on the cell surface like this picture right here, or occasionally it'll be inside the cell as well. In the digestive tract, they're almost all cell surface receptors. All right, they mediate fast cytosolic responses, so we're seeing it on the surface of the cell. And that's how hormones work. You release a signal, the signal binds a target cell receptor, and that changes the behavior of that target cell. As a little bit of a clinical, shall we say, micro case study, I don't have any assignment for this, but understand that you will be potentially responsible for questions about this article. Okay, it's called the GLP-1 type drugs. GLP-1, all right, is a type of hormone. GLP-1 is a hormone that is released when you take in sugar through the digestive tract. If you were to take sugar and stick it into your vein, if you just inject it into your vein, as opposed to taking it through your mouth, most people would believe that you would get a stronger response by injecting the sugar into your vein. That's not true, because if you inject it directly into the bloodstream, you do not get release of certain types of hormones, and one of them is a hormone called GLP-1. Now, these drugs mimic GLP-1, Ozempic and Mounjaro, and they are exploding on the market right now because they induce satiety, like I was talking about in this previous slide. So I'm gonna post this article in Brightspace, and it is not a difficult article to read, but you should take a couple of notes on what's going on, how they work, and so forth, and be able to just answer a simple question or two about the article. Okay, so starting up at the very, very top, the oral cavity, and we're going to include the esophagus. The esophagus is mostly a conductive system, but nevertheless, it is part of sort of the larger oral cavity region, if you will. So what do we have going on in the oral cavity? We certainly have glands that secrete this enzyme called salivary amylase. It does break down a lot of sugars, but it really goes after the longer carbohydrates, the starches. Okay. Carbohydrates longer than 3 or 4 or 5 individual monosaccharides. It really breaks those down. Coupled with the mechanical digestion, all right, that is the chewing, the lubrication in terms of the liquid added to the food, and then the pushing that bolus down also contributes to the mechanical digestion, if you will. Many people do advise that you try to slow down your chewing a little bit. What I mean is chew just a little bit longer. Now, there have been schools of thought that you should chew for a really, really, really long time. In fact, about 75 or 100 years ago, that was the thing. Chew, 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 chew. But we've kind of gotten away from that. You want to chew a decent amount, 30 to 45 seconds if you can, depending on the food, obviously. But just swallowing things quickly, the term is wolfing it down, is also not very good for you. So the key is a decent amount of chewing to mix with the saliva, break down those starches, start to create smaller pieces, and then emulsify as you swallow. So okay, so salivary amylase is the big one here. Mechanical digestion and emulsification through peristalsis is going on. Now we're into the next region, which is the stomach. All right, the gastro and gastrointestinal. There are a bunch of glands in the stomach. We're going to kind of break some of these down. What you're doing is you're creating a mixture of enzymes and hydrochloric acid and saliva and secretions called chyme. So chyme is not a uniform thing. It can vary depending on the type of food that you eat. When you vomit, you are basically observing chyme. Okay, that is what that is. It's emulsified and chewed material with a mixture of enzymes and secretions. So we have four major cells, and then what is their sort of primary function, if you will? 4 cells in the stomach. We have the gastric cells, and they secrete something called gastrin. All right, so it's a hormone. Then we have ECL. We're not going to say the whole phrase. It's enterochromaffin-like, okay? We have ECL cells. All right, we'll talk about those in a little bit. We have what are called parietal cells, and then we have what are called chief cells. So these are the 4 cells, and again, you should be able to recognize these cells as being part of the stomach. Gastric, ECL, parietal, and chief. We're going to break these things out a little bit. So, how does the stomach do its thing? Well, when we introduce food into the mouth and begin chewing it, and again, chewing it, decent amount, not too much, not too little, that act will stimulate the cranial nerve 10, which is known as the vagus nerve. That's part of your autonomic nervous system. Will stimulate the gastric cells to release an enzyme called gastrin. And gastrin will prep the stomach and the rest of the digestive tract in order to begin receiving and processing this food. So gastrin is released. When the food arrives, those ECL, those enterochromaffin-like cells, will release histamine and serotonin. Now, we've heard about histamine in terms of pain and itch and allergies and stuff, but histamine also works in the digestive tract. You might have heard about serotonin as part of the antidepressant system in the brain. Well, in the digestive tract, serotonin is involved in digestion, and that's one of the major secrets of human physiology is that the human body uses the same molecule to do different things in different places in the body. So ECL cells, histamine, serotonin, and those will trigger, first of all, release of acid, hydrochloric acid, via this proton pump of the parietal cells. So we have gastrin from the gastric cells. We have histamine and serotonin from the ECLs. We have secretion of stomach acid from the parietal cells, and that stomach acid is going to do a bunch of things. The two things that we're really most concerned about with that stomach acid are denaturing proteins by virtue of the acidity, and then that acid will also aid in absorption. I've got a figure here I want to show you. Now, you're not going to be responsible for all of these things, but it is good to understand that this stomach acid isn't just there to denature proteins. It kills pathogens. Certainly helps break down the food, especially proteins. All right. It also acts as a stimulant. Okay. That stomach acid actually sort of has a sort of hormone-like behavior. Certainly affects other constriction of the digestive tract, closing the esophageal sphincter, opening the pyloric. Again, all of these things, we're not going to get into all of these. You're more responsible for the stuff on the left, but understand that it goes to a deeper level, and it's pretty interesting stuff. The fourth step is the chief cells. They release pepsin. All right. So we have the gastric cells, gastrin, ECLs, histamine and serotonin, parietal cells, gastric acid, and the chief cells secrete a protease known as pepsin. All right. And you can use this figure right here. So the stomach acid causes esophageal sphincter closure and pyloric opening. Lots of stuff going on. It's really, really cool when we think of acid as just being this thing, when we can see it does so much more. So this slide basically summarizes the last couple of slides for you. We make chyme. All right. This is a combination of the food bolus along with the secretions that we secrete in order to digest that bolus. Again, gastrin from the gastric cells, histamine and serotonin from the ECL, enterochromaffin-like cells, protons to make gastric acid from the parietal cells, and the protease enzyme pepsin from the chief cells. Our next big region is the small intestine. They say intestines plurally. Oftentimes they talk about the duodenum as its own organ, along with the accessory organs for the small intestine. That includes the liver, the gallbladder, which is really a storage gland, and then the pancreas. So what does the liver actually do? We know about the liver and so forth. We might know a little bit about its function. Let's just break it out a little bit. Well, the first thing, in terms of the digestive function of the liver, the digestive function, it makes a material known as bile. All right. Bile actually inactivates stomach acid. It's a little bit on the alkaline side. The liver also handles a molecule known as bilirubin, which is basically metabolized hemoglobin. So bilirubin is the byproduct of breakdown of red blood cells. And in order to keep that iron from being reactive, we keep it trapped in the molecule called bilirubin. So bilirubin, along with cholesterol, which we've heard a lot about for dietary issues, we use bilirubin and cholesterol, and that's part of what makes up bile. Bile, again, has all sorts of things in it, but two of the most important constituents are cholesterol and bilirubin. Now, bile can and is stored in the gallbladder. The problem is that the gallbladder occasionally will develop crystals called gallstones, and if they're bad enough, some people will have to have their gallbladder removed. So you don't have to have a gallbladder. It's nice to have it if it's functioning properly, but it's not an essential organ other than a storage organ, if you will, for bile. The liver also, that bile from the liver also acts as an emulsifier. All right, an emulsifier for fats. Now, what does that mean? I've got a moving image here I want to show you. So we're going to start here. We have a fat droplet in red, and then that fat droplet gets broken up into a whole bunch of little small molecules, fat droplets, and then those fat droplets get surrounded by phospholipids. So that's what you're seeing. You see a big red circle, that's the fat droplet, and then it gets broken up. That's the emulsification. And then we surround the smaller droplets of fat with phospholipids for absorption. That's what emulsification is. I'm not sure that when you're seeing the video, it's lining up with my voice. But it starts off where you have lots of little blue dots surrounding a big red one. Then the red one gets broken up. Then we zoom in to the little droplets and surround them with phospholipids. That's what emulsification is. I mentioned this a little bit earlier as well, in that we often think of urine as the major way that we get rid of drugs from the body, and that is true. We filter and secrete a lot of byproducts from, like drugs and so forth, through the urine. But we don't give what we call the biliary excretion route enough credit. That is the other major way that we remove drugs and other potentially harmful compounds from the body is through the bile, and then the bile into the digestive tract, and the digestive tract out the fecal excretion. So it's called the biliary excretion route as opposed to the urinary or the renal excretion route. So the liver is removing two quote unquote bad things. Now, cholesterol, if you have too much of it, like anything else, is a problem. So cholesterol does some good things, but in terms of our overall diet, since we generally consume a lot more cholesterol than we need, we kind of consider it quote unquote bad. And bilirubin, now not necessarily bad either, but we don't want too much of it, all right? So we can use these as renewable resources to do other things. That is to say, the bilirubin and the cholesterol and the bile can help emulsify fats, and it can help excrete drugs and so forth. But let's talk about bilirubin for a second. If you are not properly excreting bilirubin, and the most common reason is some kind of liver issue, whether it's cirrhosis or hepatitis or inflammation, et cetera, you get what is known as jaundice. Right? Jaundice is where you have so much bilirubin circulating in your body that it begins depositing in the skin and in surface areas of the body which are visible, like, for instance, the sclera of the eyes. This is particularly useful if you have somebody that is a very dark complexion. If they're of light complexion, you can often see the excess bilirubin depositing under the skin. That's true not just with bilirubin. All sorts of compounds will do the same thing. So here's a person of darker complexion. Again, you notice the eyes are have a yellow tint to them, that's an example of jaundice in a person with a darker complexion. Jaundice is actually not uncommon in newborns. That is to say, you will see jaundice, but it is not cause for alarm unless there's a rapid onset or it lasts for more than 2 weeks, etc. There are some other tests in there, hyperbilirubinemia. Again, it's not a huge emergency, but it's something that if it doesn't go away on its own, could be a sign of something a little more serious. So don't panic if your child has a little bit of jaundice. My daughter had a little bit, and then the OBGYN said just set her out in a little bit of bright sunlight for a little bit, and if it doesn't clear up, then call me. But if it should just clear up, and that's exactly what happened. So, about the pancreas. Now, the pancreas contains two fundamental functions. One is the endocrine side, which the things like insulin and glucagon and that sort of a thing. That's the endocrine. And then we have the exocrine, which is more involved in the digestion side of the equation. And the pancreas is all about enzymes. So we have pancreatic amylase, and if you remember, salivary amylase breaks down carbohydrates into simple sugars, breaking down larger carbohydrates into smaller ones that can then be absorbed. We also have a protease. Proteases are enzymes that break down proteins, and this one's called trypsin. We have lipase, which breaks down lipids into glycerol and fatty acids. And we have phospholipase, which is different from lipase. This one actually breaks down phospholipids into the head groups and the fatty acids once again. Now that we've got the accessory organs done, now let's talk about the small intestines themselves. Certainly, the small intestines are about absorption. They are the primary absorptive surface for the body as food comes in, and so a whole bunch of things here. But let's talk about some other things. We'll get to absorption in just a moment. Certainly communication. So the big hormone here is CCK, which stands for cholecystokinin, and you don't have to spell that out. We're just going to say CCK. Once fats and protein molecules start entering the duodenum, which is the first major structure in the small intestine, you begin to get release of cholecystokinin into the bloodstream. Now, what does that hormone do? First of all, it starts to tell the stomach to slow down and stop. You've digested the material enough. We can use the digestive enzymes from the pancreas at this point. So when we think about hormones, we often think about it turning on functions. We don't often think about it turning off functions. And CCK will literally inhibit the release of stomach acid and gastrin and those other hormones. In addition, CCK will go to the brain and actually give you a sense of satiety. That is that sense of fullness, that sense of satiation. You are satisfied. And so it will help you to stop eating so you don't eat too much. Thirdly, CCK will kick in the liver to produce bile and the pancreas to release the pancreatic juice, which has the enzymes along with bicarbonate, which will neutralize the stomach acid and do some other things. So the CCK turns on the liver and the pancreas to do what they do, as we talked about previously. And then now we're into absorption. So certainly water is important, and all of these nutrients that we have down here are quite are the majority of which are absorbed in the small intestine. In terms of chemical digestion, we did talk about the pancreatic enzymes, but in the duodenum, where this is where a lot of that fat digestion is really occurring. We don't do a ton of it in the stomach, and so those lipases and those phospholipases from the pancreas are where you see a lot of fat digestion going on, where we're breaking down the fats and we're emulsifying them so they can be absorbed, or we're breaking them down into fatty acids and glycerol so they can be absorbed, etc. Now that we've talked about the function of the small intestine, let's talk about some things that can sort of let's do some extensions on this. First of all, remember that when we do some digestion here and then it enters the small intestine, and then the small intestine secretes cholecystokinin, which is a satiety hormone, right? It's a satiety along with some of those other ones that we talked about that in that extra slide. Well, if we eat too fast, you'll eat too much. You know why? Because you're only sending stuff to the stomach and it hasn't entered the small intestine. So that satiety hormone or those other ones, those GLP-1s and ghrelins and those things, don't have a chance to be released. So you'll often eat too much if you eat too fast. This is also one of the reasons why you should chew your food and try to savor it a little bit more, because that will give some time in order for those satiety hormones to be released. If you eat too much and too fast, it'll actually slow your metabolism. Now, we're in Thanksgiving week this week, and I want you to sort of notice that. If you have that big, huge meal, now my family tends to eat Thanksgiving meal in early, right after, just like a late lunch, if you will, rather than a dinner. It will actually cause you to kind of fall asleep. It slows your metabolism down a little bit. All right. Some foods, especially protein-rich foods and foods that have a fair amount of lipid in them, will make you feel fuller longer. And that's one of the hazards of vegetarianism and veganism in some cases, that people will do that in order to try to lose weight a little bit. But if those foods are primarily carbohydrate, starch-rich, which is what plant material primarily is, it won't make you feel fuller longer. All right. So a balanced diet is better at doing this. And again, I don't want to knock, vegetarianism is fine. But if you're going to be a vegan or a vegetarian, the key is to have a balanced diet that includes a decent amount of protein and fat. Not a ton, doesn't have to be a ton. You don't have to go crazy. It's that sort of Goldilocks principle. Not too much, not too little. But there are foods that will make you feel fuller for longer, and carbohydrate rich foods don't normally do that. Now we're into the large intestine leading through the colon and then the cecum and then the rectum. So the major thing that the large intestine is responsible for is absorption of water. It is fundamentally a structure that was designed to be a desiccating organ, if you will, for adapted for purposes of life on land. So absorbing water is a major aspect. Yes, electrolytes and vitamins are also important. Certainly, vitamin K is right at the top of the list, but water is the driver of the absorption of these other things. It certainly has bacteria in it as well, just like the small intestine does, and more bacteria than the small intestine has, like something like 400 known, perhaps, species. The large intestine is such a specialized environment that many of the species that we know of in the large intestine can't be cultured outside of the large intestine. That's how tight the synergism is between the bacteria and that environment. So we're thinking estimated around 400. It could be more. Like I said, vitamin K is a big one. That's because the bacteria in the digestive tract do produce vitamin K as a byproduct of the material that enters the large intestine. Folic acid is also one that's also created, but often in cases of high metabolic need, like pregnancy, the amount of folic acid generated by the fauna in the large intestine is not sufficient. Folic acid is an important vitamin for DNA synthesis. So during pregnancy, that's even more important. So yeah, these two, vitamin K is often a clotting factor, folic acid's for DNA synthesis, but those are both produced to some degree in the large intestine. Also, finalizing some lipid stuff, you can make some short fatty acids that can be absorbed or utilized as part of the emulsifier, creating the fecal material. The last piece of the puzzle is that yes, this area is a very suitable area for growth of bacteria, but it would also be a potentially good place for pathogenic bacteria to grow as well. And so the commensurate species that live there also serve as a sort of an immunological barrier, if you will, a biochemical barrier to potential pathogenic activity, whether it's coming from above through the small intestine or accidentally through some other direction. This chart here summarizes all of the major enzymes. Remember that you have enzymes and you have hormones. Those are kind of the two sort of laundry list things that you're dealing with in this chapter. So you've got 1, 2, 3, 4, 5, 6, 7, 8, 9 enzymes. You probably have a little bit less in terms of hormones. So you have the carbohydrate digesting enzymes, you have the protein ones, nucleic acids. We didn't talk about those all that much, but they're the pancreas, and the lipases for fats. So that's a nice little summary slide for you to augment what we talked about in the different regions. So this concludes chapter 26. As always, if you have any questions about what you need to know and so forth, please feel free to drop me a line, and we will see you next week when we're back in class for our final week of class. So long.